Welcome back, everyone. Uh, the first speaker of our session is Rajesh Ganapathy from JNCSA. He's going to talk about Confluent monolayers of cell mimics. Over to you, Ganesh.
Thank you. The outset, I'd like to thank Ranjini and Mahesh for this invitation to be part of this meeting. So what I'd like to tell you today about is the video that's playing in this background here. Um, and we've been bold enough to call them cell mimics. And what I'd like to convince you over the next 45 minutes or so is that this name is probably not too unwarranted. I'll try to show you data that shows that these systems share some properties of living cell collectives and why they are interesting and so on and so forth. Okay. So before I get going, let me acknowledge the people. So this work is done by the student of mine, Pragya Aurora, who is now a postdoc with Seth Fraden at Brandeis. She's a brilliant student. And the choice of the word brilliant is not an aftermath of the meeting that I was just at. She is really very good, okay? And she's doing beautiful stuff with Seth in folding DNA into capsids for viruses and all sorts of stuff. Okay, and so the experiments are pretty hopeless, as you will see as my talk progresses. Something that she pulled off. The simulations for the tail end of my talk, we had numerical and analytical help from Sauvik, who is a student of Saroj, who's right here. Max is somebody that we've been talking to for about 8 months now. It all started when I sent him a desperate email asking him about our data and what to do because I had a talk in the next 2 days. And he said, why don't you go start looking at this particular feature? We started doing that, things panned out. Ajay Sood is a long-term collaborator of mine. And of course, these are the funding agencies that let me do what I want to do. Okay. All right, so this is a video that probably many of you are aware of. All right, so what you're looking at is basically MDCK cells. It's from a canine species. These are on a substrate, and the bright spots that you see are basically the cell walls. So you can see them, you can resolve them in a microscope, and what you see there is that a lot of and of course the nucleus has been tagged by these red dots. And this was the work that was mentioned, I think earlier in this meeting. I forget precisely which talk. So what was done is in this system, they basically, in this particular experiment, of course, the cells are dividing as time progresses. The system is getting more dense. What they found is a bunch of very interesting stuff. Okay. There's something about the orientation. All right. So what I'm also showing you in the inset is basically the displacement fields of these cells accumulated over a reasonably long time, comparable to some rearrangement time of the cells. And what you find is these cells are highly correlated. They move in a highly cooperative manner. And these are plots that basically are telling you that if I were to look at the size of these regions, so I'm pulling out a dynamic length scale because these are clusters of these are the dynamic trajectories. So if I were to look at the size of these clusters and plot them as a function of cell density, as more and more crowding happens, you basically see these dynamic length scales grow. And of course, the velocity of these cells basically diminishes as the cells get more and more crowded. All right, and this is something that you would see in a dense colloidal liquid, in molecular liquids, in atomic liquids that are approaching the glass transition. All right, and one feature that's pretty striking, yeah.
Notice is not your work.
Yeah.
But, in this, the bottom, is there a peak at not goes?
Yeah.
Yeah. There.
Okay, I'm so happy you pointed it out. They don't mention it in the paper, but I'm going to show you data that it's there in the colloid experiments as well. I think it's got to do with the fact that when you hit the glassy state, you get to do more smaller cooperative rearrangements that are not spanning. So that's something that you. Yes. So yes, I'll show you the corresponding. So what you're seeing here are basically, please do stop me at any point that you want, are basically colloids. So you can three-dimensionally image colloids. And what you are seeing here are basically the fast and slow cells. Once again, you track the particles over suitably long times. And you basically find that these fast cells are spatially clustered. And that's not very surprising in the fact that if you're moved in a dense crowd, the only way to move is to push your neighbors around. So you're going to see this sort of cooperativity emerge. And once again, like Ranjini asked, if you were to look at the size of these clusters, of these cooperative clusters, you once again find it grows. And of course, when it hits the glass transition, which is close to 0.58, it once again drops, right? So this is the correspondence that they don't make, but probably it's because of being within error bars. But you do see this sort of features. So these systems basically seem to resemble a lot of the dynamics that comes about in these systems is very similar to what you see in passive systems, all right? The one thing about thinking about these sort of systems as being glassy or jammed is the fact that in numerous biological systems, there's quite a bit of remodeling that happens. And tissues are disordered to begin with, and after remodeling, they are still disordered. So thinking of them as being liquid and glassy, so that the glassy or the jam states give you rigidity is a useful concept here, which is one of the things that people are alluding to. And of course, I would also like the people who have a passing interest in these sort of systems to look at this beautiful paper which came out only last year, which is to do with the fact that it's an hypothesis being put forth by Fredberg. It was earlier put by Kaufman, which is that these sort of systems are at the verge of unjamming. So that the idea being that very small perturbations can tip you over and you get to rearrange quickly. So this is something that you need to have in biological systems. So that's to do with that. But one thing that you will find is that if you were to compare a passive material and and like the one that I was showing you, the biological living cell sheets, of course, one thing is that cells are active, right? So passive systems are just thermal. So these cells are active, so they have some sort of internal machinery that makes them mobile. And another key ingredient is that these systems are highly deformable, right? And you saw the huge amount of deformation that was going on at the individual cell level, right? So these are the key ingredients that distinguish this from conventional passive systems that many of us have studied in the past. And the fact that these are deformable also means that once in a while, you can basically have these cells come together and align. So they are deformable objects, they can form. So if you were to fit a line to these, if you were to find the long axis of these cells, you basically find this sort of nematic states that come about. And so the first part of my talk, what I'd like to tell you about is that what happens when you just turn on activity and when you introduce a little bit of nematicity, all right? So when you have orientable objects that are active, what happens to the dynamical slowing down in these systems? And then in the later part of my talk, I'll tell you what happens when you also make them deformable, right? The pointer seems to have a mind of its own, but I'll try to control it the best I can. So the first part is, of course, how does activity influence glassy slowing down? And the simplest thing, there are multiple models, all inspired by the videos that I showed you in the beginning, which is to think of them as hard particles, which of course does complete injustice to them, but then that's a starting point. And what these studies basically found is that there are two key ingredients. One is, of course, the persistence time. The persistence time is basically the time over which the active force maintains the same heading, or the velocity is at the same heading. And the second term is the velocity. And a multitude of studies basically identified that this persistence, which is unique to active systems, is the key parameter that governs active glass physics, all right? And one immediate consequence of persistence is the fact that you see motility-induced clustering, right? And this has got to do with the fact that if you have two particles that are headed towards each other, you have a head-on collision, you're going to stay clustered until the velocity vectors point in a different direction. So you're going to get these dynamic clusters even without attractive interactions, all right? So this is called as motility-induced clustering, and you see them in experiments. And so what activity is doing is that as you increase persistence, you're basically introducing more and more effective attraction between particles, all right? So you can take completely particles with no attractive part in the interaction potential, and still they'll form these beautiful clusters and crystals and so on and so forth, all right? And this seems to have a very, very fascinating outcome when you look at glassy slowing down. So these are passive liquids of hard spheres, okay? These are passive liquids in which what I'm doing here along what these people did is along the y-axis, they're increasing the strength of attractive interactions. So you can do that by putting in small molecules called as depletants, all right? And you get these sort of, you can now introduce an attraction between colloids. And what you find is that 0.58 is roughly where colloidal systems undergo, hard spheres undergo glass transition. So what you're seeing is if I were to take a vertical cut at this particular, at say any density around here, and I start increasing the attraction strength, what I find is I go through a re-entrant, all right? Start with the glass, I end up with the liquid, because with weak attraction, I form these small clusters, it frees up volume, the other particles get to diffuse, so I basically melt the glass. So that's here. And once again, when I go to very high attraction strengths, particles stick to each other very strongly. Once again, I become glassy, all right? So you see this re-entrant behavior, and persistence does exactly the same thing, all right? So I'm plotting for you the structural relaxation time. This is from simulations by these people. I'm plotting for the structural relaxation time as a function of persistence at constant density. And what you find is the relaxation time, basically some value here, dips and then goes up again, all right? So even without attractive interactions, you basically see the same sort of physics play out as you would have in hard spheres with sticky interactions, all right? And of course, all of these studies were done for particles that were, much of the simulations done for particles that were spherical in shape, and for reasons that I just showed you, what happens when you have elongated particles, right? So when you have elongated particles, the thing that happens is that you will see that these sort of defects come about, all right? These are topological defects, but topological, all I mean is that if you were to basically go around a closed loop, these are called disclination defects because you encounter an angle deficit, unlike a dislocation defect where you would encounter a displacement deficit, all right? So these are angle deficits, and there are two sorts of defects that you get. One is called a plus half defect and a minus half defect, and you will see that around the plus half and the minus half, the orientation field is quite different, okay? So there is an asymmetry here, whereas this minus half defect has a more symmetric field, orientation field, which means that the plus half defect is going to be mobile in active systems, and this is what was found. So, and in fact, what people have shown is that in living cell sheets, if you have these sort of plus half defects, that's exactly where cell death happens. So these plus half defects basically exert a stress. This is where cell extrusion happens. This was a study that came out a little while ago. And what I'd like to do is, what we were interested in asking first is what happens when you introduce this anisotropy to particle shape and ask what happens to glassy slowing down, right? So the experimental system, I'm going to go through this pretty quickly. I see that I'm running out of time. The experimental system that we're going to consider is active granules, system that has been extensively used in the past. And what we do is we basically we basically use granules and we're going to put them on a shaker and we're going to vibrate them up and down. And what many studies in the past have shown is that if you have granules with an asymmetry in a mass or in the friction coefficient, you vibrate them, you can make them self-propelled, all right? And this is something that people have been using. This is the Dosho group, this is Sudh and Ramaswamy's group in IIC. And in all of these, the essential idea is to play with shape, mass, and friction asymmetries, all right? Except now, if you want to play with the persistence, which is a key parameter for probing this glass physics, in these systems, you typically have to change the particle shape or the size of the particle, something like that. And that is something that we cannot do, all right? So because that itself can alter the glass physics. So what we're going to do is we're going to print granules, except that during the 3D print process, we're going to print ellipsoids for the reasons I just told you. We're going to print particles which have an asymmetry in friction. So you see this white portion of the particle which scatters more light because it's more rough. And we're also going to put a hole during the print process, all right? So we have a mass asymmetry and we have a friction asymmetry which we can just print. We can print thousands of these particles in one go, all right? All right, so that is just to do with the size of these ellipsoids that we work with. And of course, we're going to put them on a plate. This is the top view. We're going to image a central portion of the plate away from the boundaries. And this flower-shaped boundary is just to re-inject particles. This parameter here is the area fraction, which is my control parameter in these sort of systems. I can print thousands of particles, make them very small, and so on and so forth, all right? But to play with persistence, what I'm going to do is basically keep the particle shape constant. All I'm going to do is move this whole position along the long axis, all right? I'm changing my mass asymmetry. And the hope was that this is going to change my persistence time, right? So let me play you videos. And so this is activity 1. So every time you see a hole in the center, that's activity 1. So these particles, as you can see, so these are the trajectories. They kind of self-propel. They have this long persistent, reasonably long persistent paths. This is activity 2, and this is 3, 4, 5. All right. In these experiments, I'm keeping all of the parameters of my vibration frequency, everything constant, I'm still getting to do this, okay? So you clearly are able to play with this activity. Just to quantify this, we can look at the mean square displacement. So this is the mean square displacement part as a function of time. You go from this ballistic to diffusive crossover, and this crossover time is my persistence time, all right? And this is just the distribution of speeds as you increase, so as the colors basically are different activities going from A1 through A5, and you basically see this sort of run and tumble dynamics come out, and of course the velocity also changes. So both the persistence time and the velocity of these particles can be tuned, all right, keeping the shape constant. So the goal is that what happens, do we see the sort of re-entrant behavior that people were anticipating in simulations? Do we see that here, all right? Just to first convince you that in fact we see clustering even without attractive interactions, okay? This is the activity mediated clustering I was talking to you about. So this is activity 1, activity 3, and activity 5 at constant density of 55%, and you basically see that you form these clusters which are reasonably long-lived, all right? And we can basically look at the intermediate scattering function to basically get the relaxation time out. And what you find is that, so we basically pick a particular wave vector q, which corresponds to about the particle size, the inverse of the particle size. And if you were to get the relaxation time out, so this for 55%, we can basically now get a phase map of the, as tau p is my control parameter, my activity. This is my phi, and basically I have my tau alpha being represented as a color map, all right, which I get from my intermediate scattering function, all right. The first thing that you see is, of course, the following. That is, if you were to take a vertical cut, somewhere here, you'll see that I basically speed up in between, all right. So the relaxation here is much, much faster than on either side. So I do see a re-entrant behavior. Like that's what I anticipated in the simulations, all right? And of course, what we also do is, you see, I have ellipsoid-shaped particles, so I should in principle look at both the translational and the orientational degrees of freedom, but I'm just showing you the translational ones here. And we basically see even a mild re-entrant, so you can basically get the glass transition lines out by fitting it to sort of an MCT-inspired power law, and you basically see a weak re-entrant both in the translational and the rotational degrees of freedom, all right? This is something that was seen in the simulations when we were able to see it in the experiment for the first time, all right? All right, so now the thing is, so what these videos are showing you is basically relaxation processes. These are the displacement maps color-coded. So if you see something that's very red, it's relaxing. The velocity, the displacement vectors are very large. So it's basically large cooperative rearrangements going on here. So what you see is that if I were to look at around, say, phi equal to 80%, right? This is a pretty big phi, all right, pretty large phi. For example, for colloidal ellipsoids of the exact same aspect ratio as the one we are working with now, these are passive particles, the glass transition area fraction is around 78%, all right? Here I'm higher than that, and I still see this large-scale cooperativity come out, all right? Now the question is, why does this cooperativity come out, all right, and what's going on in the system? And yeah, so what we do is that I basically told you that in these sort of active ellipsoids or active elongated particles, you basically have these sort of defects which can be motile, all right? So what we do now is to basically identify these plus half and minus half defects. So you can see basically the comet shape here and the trefoil shape here. The colors are basically the orientation vector fields, orientational fields. And if I were to play this movie for you, what you'll find once it decides to play is that the minus half is more or less stationary, whereas the plus half is mobile, all right? And this is not just true for this particular instant in the video. We do it for various patches, for various instances of time. And the dynamical heterogeneities, which I was pointing out, so you can basically identify the fast and the slow particles by just calculating the displacements. You calculate the Van Hove function over a reasonable amount of time, which is basically the cage rearrangement time. The central part of the Van Hove is particles that are not moving at all, so they are the central part of the distribution, of the displacement distribution. The tail end are the ones the particles are moving quite a bit. We can basically look at these particles which are colored in red, and these dynamical heterogeneities are precisely in the regions where we basically see these plus half defects moving, all right? So what all of this basically tells you is that you have a very, very strong link. So this is the plus half and minus half defects that we see here are just something that you can pick from a snapshot. You give me a picture of the ellipsoids, I look at the orientations, I can pick this plus half and minus half defects, and what we are showing here is that there's a beautiful correlation between dynamics and the orientation field, all right? Something that's very, very tricky to do with, right? So, so far what I've told you is basically that these sort of defects interfere with glassy slowing down. Okay, a system that should have been glassy had been passive is actually motile and in fact showing large-scale swirly motion. But then this system is an oversimplification of the video that I started off with. Okay, yeah.
And minus half, and why one of them would move and the other would.
Oh, yeah. So the plus half has got a more anisotropic orientational field. And the minus half has got more, it's more symmetric. So the vector, so there is an active stress that couples the orientation field. So an anisotropy there is going to get manifested as a motility.
The way they're packed is different. The plus half.
Yes, the way the particles are packed around the minus half and the plus half is very different.
Okay.
Yeah. All right. All right. So we still have one of this oversimplified system. We don't have anything that's even mimicking what we saw in the video for the first time. All right. So what we want to do is to bring in deformability as well. All right. And I'll tell you why deformability is fascinating in these systems. And I'll start with this video that's playing. It's going to loop, so you're going to see it again and again. So basically, this is a Drosophila wing that's developing. All right. And if you look at the early stages of the video, you see there is a lot of size change of the wing also taking place. But if you look at the early stages of the video, what you find is that this is a system in which there are no free voids present. It's a completely packed layer of cells, and yet this system can go from being rigid to being fluid. All right, I am not controlling density here. But the density of this assembly of this confluent layer is fixed. All right, so and this is something that happens in numerous biological processes. For example, when cancer cells grow from within the tissue, right? They're growing in a fully crowded environment, and yet they are motile and they move to other parts of the body, right? Oh, there is, the flow here has got to do with other signaling pathways and all of that that's wanting you to form this final state. But the one that I want you to focus on, and I'm not too aware of that, the fact, what I'm only trying to say is that you have a completely packed layer that can change morphology and then become solid or fluid, right? And this, of course, plays a role in numerous other things, including wound healing and organ development and all of that, all right? Now, in fact, there is a bunch of models that capture this sort of behavior, and these was mentioned in numerous talks before, and this is the vertex model, okay? And this is for 2D sheets, all right? First developed by Farhadi Far and then other people. I think the first one is by Honda and others, all right? So what you're essentially having in these models is the following. So you're basically looking at a perspective view of the cell sheet, and this is a top view, all right? Now, what people find, and each of these terms in the energy function that I have written is motivated by experimental observations, all right? The first term, of course, is the fact that when you have these cell sheets, you know from observations that the cell volume, of course, is conserved, and you find that the height fluctuations are not much. Okay, the cell height fluctuations stay more or less put, which means that any fluctuation in the area of the cell, if you were to look at it from top, you're going to pay a penalty, so which is this term here. So you have this area stiffness term here, all right? The last term is got to do with the fact that every cell has got this actomyosin cortex inside, okay, which gives it some sort of stability. And if you want to stretch it, okay, if you want to stretch it like a spring, you're basically going to change the length, and you have something that you put a penalty for that as well, all right? And the term in between is a competition between two effects. One is the fact that you would like to maintain a circular shape. It's got to do with the line tension that's linear in the length. And the other thing is, of course, that if cells like to adhere to each other, then they would want a longer length, a longer interface between them, all right? So this term, this lambda ij can be either positive or negative, all right? And now what was found was pretty remarkable in this model by Max B and Lisa Manning in about 7-8 years ago, which is that these sort of systems, there's actually a transition from a solid-like state to a fluid-like state just by changing the cell shape. So you can have a shape index, so this is called as shape index, which is basically the perimeter by the square root of the area. Yeah.
Question.
Yeah.
Is there any specific reason for the last term, that atomising contractility, to be proportional to perimeter square? Like, could it
This
be
is basically
perimeter?
like stretching a spring. It's like stretching a spring. So it goes as quadratic in the length.
So you're assuming that equilibrium perimeter is 0, like L minus L0 whole square. Could it?
The equilibrium, so the cells have a preferred perimeter.
Yeah, is
The
that
cells have a
0?
preferred perimeter. Around that, if you were to extend, you're going to pay a penalty for extending.
Yes,
that.
yes.
So that's going to be quadratic in the length. No, I don't have. Yeah. So
That target, is that target, is it 0 or is it getting?
so basically, what I can do is I can basically club these terms together.
Yeah,
Yeah.
I see. Okay.
All right, and then round it off, and I'll have something that goes as something that depends on the ratios of these quantities and L minus L0 squared.
Yeah.
Yeah, I've just split it into 3 terms. Yeah. So what you find in this model is that if you were to look at the target index, by just changing the cell shape, you get to go from a solid to a fluid-like state. All right. What it is saying is pretty phenomenal, because it says that if you give me the picture of a cell sheet, all right, I can just tell you I can just calculate this by looking at the perimeter of all the cells, the shape index of all these cells, and if this number is greater than 3.81, it's going to be a fluid-like state. If it's less than 3.81, it's going to be a solid-like state. All right. And there's something that was seen in experiments on asthmatic cells. Okay. So what I'm plotting for you is this dashed line is 3.81, and you're looking at the maturation of bronchial cells. All right. And what you find is that if you were to plot as the cells mature, as they become jammed, as time progresses, in healthy people, what you find is that this quantity, as maturation proceeds, starts from some value that's much larger than 3.81, it's fluid-like, and then approaches 3.81 by day 8, except that if in asthmatic people, what happens is even by day 14, it's basically not reached this threshold, okay? It's still fluid-like, yeah? And one thing that will bother you, as it does to many people, is that if you look at the width of these distributions, okay, that's basically how much shape variability you see, all right? The cell shape variability is huge in both of these cases, all right? And I'll come back to this point a little later, all right? So cell shape basically plays a very big role in how systems go from solid to fluid-like states. And in fact,
So I just wanted to ask whether the volume fraction in the previous case where the P is changing, it remains the same or it changes?
okay. So in one of the reasons we did the experiments is because in real systems, I'll come to that now, is that you always cell death. So these are experiments that are tracking cell lines over 14 days and 8 days and so on and so forth. There is cell death that goes on, cell division goes on, all right? And if you are at the verge of jamming, you don't need to change the density much to push it either way, all right? But the models are confluent. So there the packing fraction stays at 1 throughout. Yeah. Yeah. Okay. And whatever I showed you was for 2D sheets. And in fact, recently this work that's come out by Joseph Kass's group only this year, in which you basically find that this sort of connection between cell shape and cell motility is there even in 3D. These are basically breast cancer explants, and you basically find that regions, not at the system level, even if you were to look at it locally, you basically find that regions that are more motile are basically comprised of elongated cells. So they are slightly more longer, right? And this is basically just the time lapse, all right? Yeah.
Here the cells are not confluent, right?
Oh, you're looking at this is just a few cells that have been tagged.
Oh.
This is a dense tissue, it's a 3D tissue. And I think this is tracking by hand because they find it very hard to do it in 3 dimensions. Yeah. Okay, so the question, of course, is something that you were asking a little while ago: is this jamming and jabbing purely shape-driven? Because in these systems, of course, all of these other processes go on. And it is not just mechanics, cell can talk beyond mechanics as well. And all of these shape-driven transitions, is it unique to living cells, living collectives, or can this also be mimicked in synthetic systems? And that's going to be the rest of my talk, all right? So what we want to do is we want to make these cell mimics. We want two ingredients, of course. One is activity and the other is deformability. And the way we're going to do it is the following. We are not the first to make deformable active matter. So people have made deformable active systems before. And for example, these are granular robots that are confined within metal rings or paper rings. You can look at the size scale here and you'll see immediately there's a coupling between orientation of the robots and basically the shape of the ring. They tend to pile up at these corners because the orientation gets coupled to the velocity vectors of these robots. Okay, that's, of course, pretty huge, 10 centimeters. So if you want to make a confluent assembly of this, you're going to look at something that's the size of this floor, all right? If you want to look at a few hundreds of these cells, all right? The other one, of course, is you can now take vesicles and put active colloids within them. And these are studies, but to the best of our knowledge, what we haven't seen in the literature is cell collectives of deformable active particles, all right? And that's what we want to do. And so what we do is we take the ellipsoids that I was just showing you and we put them inside paper rings. And so these are achiral particles, right? They just had a motility, right? And if you put them inside paper rings, what you find, of course, is what the robots did, all right? Right? They pile up at the corners, except now you should look at the size scale also. It's about a centimeter, right? So it's doable in experiment. I can now make a few hundreds of these cells and put them inside observable area. But one thing that you do find is that these particles pile up at the boundaries, and that's simply because of the fact that there's a persistent active force. So when they come to the boundary, for the same reason that they form motility clusters, they're also going to pile up at a boundary, all right? Now, if you don't want these particles to pile up at the boundary, what you need to do is to get them there and tilt, right? And so you need more than a force, you need an active force and an active torque, all right? Now, the way we made these particles active was putting a hole along the major axis, but then what we can do is we can now just shift the hole along the minor axis, all right? So now what happens is you have two forces, one that's, and they're not congruent, they're not coincident, which means that these particles, when you put them on a plate and vibrate them, they basically undergo circular trajectories, right? Now, and because they have these circular trajectories, when they come near a wall, they're basically going to hug a wall. And depending upon how you place these particles on the plate, if you take these left-handed if you take these particles which like to make anti-clockwise turns, they basically hug a left wall. And if you take these clockwise particles, they basically hug a right wall, all right? And of course, what we're going to do now is to take these chiral particles and put them inside our membranes. By n ensure pure, I just mean that all particles inside have the same chirality. It's a pure, it's all of one chirality, which is what I'm calling by n ensure pure. All right. And now let me also play this video. Okay, so these are particles which are just going around in circles, and you can see them actually embrace the wall, right? And all this particle does is basically spin, all right? Because all it has at the end of the day is an active torque, all right? Now, to make this cell motile, what we do is the following. To make this membranes motile, what we do is the following. We flip a few particles so that they have the opposite handedness, all right? So there is going to be this current that goes like this and this current that goes like this. The net current is going to be somewhere pointing up, all right? And when I play you the video of this, you will see that this is quite motile as compared to this one, all right? Now, what it also means is that by just playing with the number of particles that I flip inside these membranes, I can basically control the motility of these cells, all right? So this is the least motile and this is the most motile, and I can just get all sorts of motility by just changing the net chirality of the cell interior, all right? And that's what the video is showing, all right? So this is basically a control parameter for me in my experiments, all right? Yeah.
And achiral particles?
So yes, so we really haven't done those experiments, But this is a little cleaner in the sense that all I have to do is, you see what I'm also doing here is keeping the number of particles inside these membranes fixed. All right, I'm not changing anything about the membrane. All I'm changing is the net chirality inside, and I'm able to make them motile.
No, what I was telling that inside the membrane,
Yes.
if you put, let's say, same number of particles,
Yes.
let's say you have 15 particles, so 10 may be chiral and 5
Sure.
achiral in all cases.
Sure, but what I'm saying is we haven't done those experiments, but
Okay.
so there might be some interesting physics that comes out due to this competition. But the one that we're doing is just to have a constant number of particles and still play with the chiral. All right. So this is what we see. We are able to tune this, and so once again we calculate the mean square displacement, the velocity, and then I'm plotting for you the persistence time as a function of this chi cell, which is the interior parameter, which I'm able to play with. All right, and of course I'm able to play with the persistence time of these cells, and also the persistence length, which is just the velocity times the persistence time. All right, both of them go up. All right, something that we didn't anticipate that came along for the ride was the following. All right, so let me. So what you have are these particles that are exerting a torque on these paper membranes. Right, they're trying to push. They're trying to go anti-clockwise. The membrane is stopping them, so they're exerting torques on these membrane. All right, and now if you were to look at the inertia pure and the racemic one, the racemic has got equal-handedness. You basically find that this membrane is a little more floppier than this one. Right? It fluctuates a little more, right? And we can actually fit it to an ellipsoid, and we can calculate the aspect ratio of this ellipsoids as a function of time. And then what you find is that as you change the net chirality, the standard deviation, which is a measure of how much these cells fluctuate in shape, also increases. All right? And this is going to have an interesting consequence to what we see. All right? So now I'm all set, except I have to take care of one final parameter, which is the following. I can now make this confluent assembly of these deformable membranes, right? Except that if I were to now have all of these particles of a given chirality, if all of them are right rotating, then what you find is that you see these sort of edge currents. All right, and that's something that I won't go into. This was seen in chiral active systems by Vincenzo's group a long time ago, and that's something that you see in deformable systems as well. But you don't see this in confluent epithelia. So clearly, chirality is something that we want to get rid of. So what we do is the following. For every activity, for every control parameter that I have, I'm going to have equal amounts of left rotating and right rotating cells. All right. So then I have this confluent monolayer, and this one doesn't show the sort of currents that you see here. All right. And this is the sort of systems that we're going to basically study. Yes.
But don't put it to be so dense.
Yes.
Will they form clusters?
So this is purely a boundary effect. So you need to confine them, or else you need something else to mediate an attractive interaction. Yes, but otherwise they wouldn't. No. But then we are interested in looking at the very dense limit, which is why we have to get rid of this chiral currents, which we do by mixing equal amounts of left and right rotating cells. Sure, but yes, but there was also the fluid that was mediating this interaction between two different cells.
Yeah, so you need some sort of attraction.
Yes, these systems wouldn't show. All right. So we have this. So now we are going to basically take this system. We're going to probe the dynamics at different phi's and tau p values. We're going to see how different or similar the system is to a living cell collective. All right. So this is 0.45, and what you find is that as you increase persistence, just like we saw in the passive system, you basically see this sort of clustering come about. All right. And you can basically do this clustering analysis very easily. You just have to calculate the Voronoi areas. And what we find is that as you go across an activity, you basically find that the highest activity, you have this shoulder developing, which is also telling you that you have these small clusters coming about, right? But then what's fascinating is what happens at very high density. So this is 0.92, okay? And this is the maximum number of cells that we could pack on the plate. So this is, I'm going to show you now movies for three different values of activity. This is at the lowest activity that I have. This is at the highest activity. All right, and you will immediately see that although my packing fraction is fixed, the more active one is more fluid-like. But what's fascinating is that if I were to, and of course these are cells that are stiff, so they basically take these nice circular shapes, whereas these cells are individually also floppy, all right, because of these two competing currents inside. Except that if I were to look at in-between persistence time, you basically find that this system is faster than the one even at the highest activity, all right? So what I can do is, to convince you of this, I can basically track the center of mass trajectories. You find that it's completely localized here, a little more fluid like here, but for an intermediate value of tau p, you see that's almost ergodic, all right? Now the question is, why do you see this reentrant behavior in these sort of systems? And we're once again going to calculate the 2-time density correlation function, get the tau alpha out, all right, which is the relaxation time, right? And so once again, I'm plotting for you relaxation phase diagram. So this is tau p versus phi, and this is basically, the colors are basically my alpha relaxation time, all right? Now what you find, of course, is that in this system, unlike what we saw in passive systems. For example, let me just first tell you what I see here, that if I were to keep tau p fixed and I just increase phi, I'm just increasing density, the system should slow down, and that's what I see, all right? Now, instead of that, if I were to take a vertical cut, all right, something at around, say, phi equal to 0.75, I increase my persistence time. What I see is I do not see the reentrant that I saw. For example, in the active ellipsoids but rigid particles, we basically started seeing signatures of the reentrant happening even at low density. That's not something that we see here. But what we see is a reentrant that comes about only at the highest densities. All right. So you basically see the relaxation is very slow here, very slow here, but for an in-between density, it's kind of fast. All right. Now the question is, because this happens only close to confluence, is it something that's got to do with the shape of these cells? All right. And so what we're going to do is we're going to calculate two quantities. One is the average aspect ratio of all the cells as a function of time, averaged over all cells in time. And the second one is basically the standard deviation of the aspect ratio, which is telling me how much these cells are basically fluctuating in these assemblies. All right. So if I were to calculate that, so this is once again the same phase diagram. What you find is that the aspect ratios of these cells, it is at that intermediate tau p where the system was fast, right, that you basically have the aspect ratio also to be larger. So it is at that reentrant transition that I find the cells to be more elongated, all right? And that's not just true with the cell shape, even the cell shape variability, which is basically looking at the fluctuations of these cells, both of them actually show a reentrant right at the persistence, right at the tau p at which I basically see this faster relaxation, all right? Now the reason I have the picture of Pragya here is the following, right? Each point here is an individual experiment, and each experiment has to be assembled by hand. Every cell is assembled by hand, in which he takes a cell, puts the particles of the right hand in this, and this entire phase diagram is constructed. Okay, it's a hopeless experiment as experiments go. All right. So, and in fact, if you go to densities where you do not see the reentrant, you also do not see the reentrant in the cell shape or in the shape variable. All right. So we have a kind of a picture of what's going on now. If you're at very low persistence, each cell is very stiff. It's also got a very low motility. And this activity-mediated adhesion between cells is also very small because it's got very low persistence. All right? If you go to the highest activity, each cell is very floppy. It's got very high motility. But then this activity-mediated adhesion between cells is also large. So you can imagine that when two cells come and adhere to each other due to this active attraction, what's going to happen is the membrane in between is not going to fluctuate much. Okay, and that's why the shape variability comes down. And of course, at some intermediate value of tau p, these effects compete. All right, and you see this reentrant behavior. All right. Now, the fact that the cell shape and the shape variability basically mimic each other tells you that these two quantities themselves are kind of connected to each other, right? They basically mirror each other, right? So, and this was this paper that I was talking to you earlier about, in which they found that if you were to plot the standard deviation of the aspect ratio versus the average aspect ratio for a whole bunch of different systems, cancer cells, MDCK cells, and all of that, you basically find that all of them lie on this particular line, and this line was predicted by Saroj to have this particular functional form, all right? And this scaling that was predicted by Saroj, I believe he's going to talk about it tomorrow, is basically just for a bunch of closed loop objects, all right? So which living cells are, which even my synthetic cells are, all right? So what I'm going to try and do is basically see if this constraint between standard deviation of the aspect ratio and the average aspect ratio also falls through for our experimental system. And one before I go there, what I want to also say is that as you go down this line, what the study further found was also the following. As these MDCK cells jammed, this distribution, so you basically plot the distribution of the aspect ratio, that's what this is. This distribution gets less and less skewed. So what it means is the mean moves further down the line, so does the standard deviation, which means that as you jam, you're moving down this line, all right? And of course, you can collapse all of these distributions, and this happens to be a k-gamma distribution, all right? So what we want to see is if all of this falls through for our experimental data as well. And these are our granular cell mimics, which lie on this line, and this is just the zoom in, and what I'm showing you here is that this agreement between what Saroj predicted and what we see in experiments is not just true for confluence, but also extends far below confluence, all the way down to 74% area fraction, all right? We can now calculate the probability distribution of the aspect ratio. So these are the aspect ratio distributions for various values of tau p. You can scale them as so, and they all collapse onto a single distribution, and this thing can once again be fit to a k-gamma distribution, all right? So this is all very nice, right? Now, in everything that I've done so far, all the correlations that I pulled out so far are basically by looking at the entire system as a whole, okay? And we know from everything that I've told you before that there are these dynamical heterogeneities in these systems. So you have these fast cells and slow cells, and the question is basically whether this sort of analysis also falls through for the fast cells and slow cells. So you take an ensemble and then you look at just the fast and slow, are they going to also lie along this line? All right? And what you would expect is the fast cells being more mobile should have a higher standard deviation. They should fluctuate more. They should also be more elongated, whereas the slow cells should be somewhere down the line. All right? And so this is what we set out to do. All right? And what we're going to do is basically calculate these dynamical heterogeneities. Once again, the way we do it is the way we have been doing it all along. We basically calculate the probability distribution of the displacement, look at the central part of the distribution, that's basically cells that don't move a lot, and look at the tail ends, and these are the cells that move quite a bit over this time scale t star. And then once we have this fast and slow cells, these are basically the videos that I'm showing you of what goes on as you increase tau p. So this is increasing tau p, and what you find is that like in the passives, like in numerous other glassy systems, you find that these are clustered. So once every time you see a dynamical heterogeneity, you'll see that comes about in clusters. And of course, we find that the one with the more fluid-like state, the one that had an intermediate persistence time, is also most dynamically heterogeneous. But now what we can do is from these sort of experiments, we can just calculate the aspect ratio of just the fast cells and the slow cells, all right? Which is what I'm showing you here. And what you find is, as you would expect, the fast cells of these assemblies are a little more elongated than the slow ones. But then if you try to collapse them on the line, you try to find whether they fall on this particular line, you basically find that the slow cells are basically along this line, whereas the fast cells are not. Right? They are basically below the line. So what you're seeing is that these cells are more elongated, but they also fluctuate much less. All right? The question is, why does this happen? And this is where, when we came to, and by the way, Saroj has performed these simulations with both the vertex model and the cellular pots model, and in both cases, you basically see results that are in agreement with what we see in experiments. Right? So the question is, why does this happen? And so one way to think about it is the following. So this is what we've been mulling about, and this is the final picture that we have, is that what you can do is, over a suitable time scale over which rearrangements happen, you can imagine the most mobile cells to be caged in a matrix of immobile cells, all right? because these cells are not moving, these cells are moving, so which means that this is an immobile matrix for the most mobile cells. And then what we do is that now we basically, Saroj does these simulations in which you start with a system, okay, in which you equilibrate the entire system, but then you freeze everybody else but a small subset of cells in the center, right? Now, you will immediately realize that if I had just one cell, it's going to get frozen in whatever shape it had before the freezing, and it's not going to fluctuate in shape at all. The standard deviation is going to be 0. It's going to be below the line, whereas it's going to be elongated. As you increase the system size, as you make the system size larger and larger, these fast cells are going to adhere to this line, and that's what we basically see. So these are simulations done for various cluster sizes. So these are the blue particles, blue patches that you see here. So you start with one cell, of course, there's no standard deviation, it's 0. So you start with 6, 8, 10, 12, and 14. You basically find that these cells basically collapse onto the line for large enough clusters. So when you allow big enough clusters, there's enough shape variability. All right? You had a question? Okay. All right. No, it only, no. You see, the thing is this, they still have a long enough, they're still elongated. It is just that the confinement effect that comes about due to being caught in this immobile matrix suppresses shape variability. Yes. Yes, the fast cells were not falling on the line, but they were more elongated. Yeah. Yes, they are, yes. So there is a connection between fluctuations and elongation. Yeah. Yes. No, it does, meaning these are just the top 10% slow or fast cells. This is just the top 10% faster slow cells. That simulation is an idealization in which we're taking a small patch of the system and letting it evolve, whereas the rest of the system is frozen, trying to mimic fast cells caught in the immobile matrix. Yeah. So that's where we are. All right. So this reduced shape variability is an outcome of this confinement that comes about. That's temporary. It's fleeting, but then it comes about once in a while, and then yeah. Yes. 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 They will all be along the line. They will just be various points along the line. And when you take an average over the entire system, it will be somewhere in between. So the fast cells will be further down the line. The fast cells will be further up the line, the slow cells further down. And of course, when you do a time average overall heterogeneities, it will be somewhere along the line. But what we find is something that's not the case.
For a given cluster of fast and slow particles,
Yes.
one should not expect that this variation of this aspect ratio should be larger for the faster cluster. And
the variation of the aspect ratio is, we thought it would be faster, but it is below the line, which means it is less than what it should be.
okay,
I don't know if I'm answering your question, but we can talk about. Oh, so that's basically what I'm saying, is that you have this sort of temporary confinement that's coming about. So when you confine these cells, you don't get to shape, you don't get to fluctuate in shape too much, right? And we believe that's what's causing, because when we do it as a function of system size, you basically at n equal to 14, you essentially fall on this line, right? Okay. Okay, so that brings me to the final slide. So we have this synthetic system made of plastic particles and paper. And you look at dense assemblies of these systems, and it captures many features that you see in living cell collectives. This probably, to the best of my knowledge, the first time you're seeing a reentrant glass or jamming transition that's mediated purely by cell shape changes. Um, there was this beautiful essay that came out by Manning some time ago, which is essentially about these sort of artificial systems that are confluent. And of course, with this sort of system, we have quite a bit of handle because you have the ability now to play with the stiffness of these membranes, the nature of activity. For example, one of the things that we started out doing is basically look at membranes with different stiffnesses and see if they spontaneously sort. And this is something that happens in biology. It's called cell sorting, all right? And we can, of course, change. We can just print particles of whatever we want. These are active Brownian particles. And you can immediately see the nature of fluctuations here is very different from here. And one thing that we are really fascinated with, which my student Uttam is going to probably get working, is that now we have membranes that are made of discarded magnetic tapes, which means that you can play with the magnetic fields and play with these membrane stiffnesses in situ, all right? So, and the idea is to basically look at these sort of systems as well, all right? Thank you, and if there are questions, I'll be happy to answer them.
Yeah, time for some question.
Can you quantify these fluctuations? Suppose this Q dependence of this membrane fluctuation to estimate the stiffness of the individual cell, like people do for this vesicle fluctuation and all?
We haven't done that, but that's on the cards. In fact, what we're trying to see is if the nature of these fluctuations does it encode what activity is inside.
Okay.
Right. So does the spectrum of whatever that we will see in the fluctuation spectrum, does it give you inclinations about what sort of activity is inside there? So we are trying to get these experiments going. Yes,
Yeah.
but that's precisely what we want.
Okay. Just another question. This elongation or aspect ratio, does it work for these Lisa Manning's, like where this vertex model predicted this some area perimeter, critical area perimeter ratio? Just based on this aspect ratio, can you predict solidification or fluidization, or you need more ingredient?
So the aspect ratio, the 3.81, corresponds to an aspect ratio of close to 1.18 or something like that. So in this systems, we try to see if the reentrant transition that we see happens across 3.81. We just see so much scatter within the data that we are unable to make out any clear trends. And the distributions are quite wide. So for me to draw a conclusion based on that, I'm not brave enough.
Yeah. Okay, thanks.
It seems to me, it seems that in this system, right, the perimeter is actually fixed.
Yes.
So it's just the vertex model version is just the a minus a0 squared term.
Yes.
It shows many of the same features as when you have the perimeter term as well.
Yes.
Are you able to make systems where basically you use something like very weak rubber bands instead of paper bands in order to have a variable perimeter? Because there's some things that can only happen when you have that perimeter term.
So one of we haven't done that. So and these particles exert such tiny forces that I still haven't found a membrane that can easily extend for the kind of forces that these particles exert. Bots would be better, but they're too huge. So one of the reasons that we are fascinated by this particular set of this particular system is that you don't have the perimeter term is fixed, and yet it seems to show many things that that you see in living cell collectives where the perimeter term plays a role.
Well, actually, I mean, I think if you take the vertex model without the perimeter term, you see almost the same things. You see many of the same things as if you include the perimeter term. It's not so important for a lot of the phenomena that you're talking about, just to have the area term only.
So the area term also has a very weird origin in this, in the sense that there it's got to do with volume incompressibility and suppressed height fluctuations. Here I don't even have that. I mean, these are just 2D membranes open to Yeah, probably. Yeah.
But another way to introduce something that's like the perimeter term would be to put some adhesion on the thing. So if you use, for example, post-it notes, very, very slightly sticky
Yeah.
perimeters,
Yeah.
with that would introduce something like the perimeter term, right?
It would, but then, yeah.
I can imagine other difficulties that it
Yes.
would not maintain the same adhesion over time.
I have so in one of these movies that you showed that there are some cluster formations,
Yes.
and but maybe I don't know whether it's your data or somebody else's data. And if you, I think this was
With
initial,
the deformable ones?
yes,
Okay.
the initial maybe phase of it, but in the dilute limit
This one?
regime, yes.
Okay.
Yeah, and there the thing that caught my eyes is this the structure of those clusters.
Yes.
If you look at it, they are often more rough than what you typically you expect.
Yes.
Right, because the surface tension would like to bring them to be more circular. Then do you have some feeling about it?
So the thing is this, what we find here is that the adhesion is very weak. I mean, these clusters are so short-lived that we really have to,
No, but if you go back to some others work also that you present here in
Yes.
one of these movies, there
Yes.
also you see this clusters are more elongated objects rather than spherical objects.
Yeah. So no, for the Janus colloids, you form these really compact clusters.
Always?
Yes. So, but it does. So, what happens in these systems is what people have shown is that even if you have a slight amount of torque that comes about due to some imbalance that you have, these clusters are not stable. They rapidly, yeah. So, and it is impossible in experiments to completely get rid of those torques. So, you never see. Maybe that's one of the reasons.
So that leads to more fingering in the surface.
Yes.
At which the particles are getting attached to the cluster, like the growth thread, and that can actually change the shape. Actually, like if you look at tumors which are benign versus which are malignant, you will see much more rough edges in malignant tumors which grow faster, whereas benign tumors are more like the surface is more smoother. So that can be one reason, like the growth rate at which they are getting added might be much like faster, and that can give rise to this roughness on the. Yeah, but I'm saying the growth at which it is happening is much faster. So if it is a malignant cell, it grows much faster than a benign cell, and that's the reason many a times you do not see well-formed this capillaries in there. They're bleeding cells usually. So that's the rate of formation, and that can give rise to maybe the smoothness versus the roughness there.
Looking at the.
Just if you look at the fluctuation of the total cell, you control that fluctuation with the ratio of the chiral and achiral particles and other things.
And yeah, at the level of a single cell.
Single cells, and that is the most dominating factor to the overall phenomenon also, so how that fluctuates. But what I was looking for, the number of individual particles inside a cell.
Yes.
Now that might also control the fluctuations level, and due to collective phenomena that depends upon the number of cells, not the individual particles inside the cells.
So in all the experiments that I've shown you, the number of particles inside the cell stays fixed.
Yeah.
The number of particles in the cell stays fixed. The cells do, at the individual level, they do fluctuate differently depending on how active they are.
Yeah.
Right? And all of this, plus what happens when you go to, so when you go to the dense limit, you have all this active clustering coming about. All of those effects compete to see what we see.
Okay, so that's why I was thinking like that. If it is only the number of particles controls the fluctuation inside a single cell, let's say. Now, if you know how the cell fluctuates depending upon the number of inside particles.
We haven't changed this.
Yeah,
we have kept it constant.
Yeah, that is what I was thinking. Then the same phenomena could have been reproduced with various particle concentration or not.
Sure, but yes, that's another parameter that we have which we haven't played with.
Okay.
I just one more question. Sorry, those are beautiful experiments, by the way. That's very cool. One thing that people haven't looked at so much, but that could be interesting, especially in the case of Yosef Case's systems, right? The cancer explants. What if you have heterogeneity of motility, right? That's something you could do really easily.
We did that.
You did that.
We did that. So despite what Pragya went through, she still agreed to do those experiments. So we did have identical membranes, but different activities. We wanted to see sorting or phase separation, something that comes about. We have 124 cells on the plate. That's all we could assemble on a reasonable time scale. We didn't see anything over a day of experiments. But then we wanted to play with the stiffness, keeping activity constant. We wanted to and see if that comes out, but we haven't done those experiments. Yeah, but maybe more distinct levels of activity, maybe things can happen. Yeah, more wider, because we just have to change the ellipsoids that we put in. Yeah.
If there is no more question, let's thank our speaker again. And it's time for coffee.